Hi, I'm Dr. Christian Bogner from Autism is Biomedical. For decades, autism has been explained through behavior, genetics, and vague environmental triggers. But what if we've been missing the most important piece of the puzzle, an unseen chemistry inside the brain itself? Today, I want to introduce a radical new hypothesis that endogenous psychedelics, indole tryptamines such as DMT and Bufotenin, may be central to the autistic phenotype. These compounds, long thought to exist only on the fringe of human biology, or in the deep Brazilian rainforest, are produced inside of us. And if they are dysregulated, they may alter perception, sensory experience, and cognition in ways that could explain autism far more directly than any model before. This is not a metaphor. This is biochemistry, and if true, it could redefine everything we thought we knew about autism, shifting the conversation from mystery to mechanism, and from mechanism to meaningful interventions. To move this bold hypothesis forward, we are not working alone. We're actively collaborating with some of the brightest minds in the world. At Stanford, we're working with Dr. Mike Angelo. Dr. Angelo is a leader in high-dimensional imaging, and he has his own laboratory, the Angelo Lab. At UC Davis, we work with Dr. David Olson, who's the director at the Institute for Psychedelics and Neurotherapeutics, and we work with Dr. Stephen Barker at Louisiana State University. He's the world's foremost expert on endogenous DMT research. Together, we're designing a study unlike anything attempted before, the first comprehensive measurement of the entire psychedelic metabolome in autism. This has never been done in autism, and in fact it has never been done in any psychiatric disease. For the first time, we are preparing to quantify these indole tryptamines, serotonin, tryptamine, bufotenin, DMT, and beyond, in the context of autism, to see whether dysregulated psychedelic chemistry could explain the striking neurologic phenotype we observe. So let's clarify what we're talking about. These are the indole tryptamines, serotonin, tryptamine, DMT, and bufotenin, all derived from the amino acid tryptophan. Serotonin is the neurotransmitter everyone knows, critical for mood, sleep, and sensory integration. Just one enzymatic step away is tryptamine, the backbone for more complex molecules. Add methyl groups, and you arrive at DMT, one of the most powerful psychedelics known. Add a hydroxyl group and a methyl group, and you have bufotenin, another psychoactive compound made inside of us. Here's why this matters. Structurally, these molecules look strikingly similar to LSD and psilocybin, the very compounds that can radically alter perception, sensory gating, and cognition. Even ketamine, though not an indole, shares overlapping receptor effects on glutamate and serotonin systems that intersect with these pathways. In other words, autism may not only involve an imbalance in serotonin, but a dysregulation of an entire psychedelic biochemistry that mirrors the same class of molecules capable of inducing altered states of consciousness in humans. That is why studying them is not just fascinating, it may completely reframe how we understand autism. Before we dive deeper, let's ground ourselves in what DMT actually is. N,N-dimethyltryptamine is a potent psychedelic compound that exists not just in plants and in animals, but also in humans. It is capable of producing profound alterations in perception, cognition, and even motor function, exactly the domains where autism presents some of its most striking features. In the brain, DMT acts at multiple levels. On the molecular level, it binds to serotonin 5-HT2A receptors, the same target as LSD and psilocybin, and also to sigma-1 receptors, which are involved in cellular stress regulation and neuroprotection. At the network level, DMT radically alters brain connectivity. EEG and functional MRI studies show a collapse of rigid patterns and an opening of crosstalk between regions. On the perceptual level, it changes sensory gating and predictive processing, essentially reshaping how the brain filters and interprets reality. This framework gives us a way to think about autism in a new light. If endogenous psychedelics like DMT are dysregulated, then the alterations we see in sensory processing, cognition, and motor control could emerge directly from this chemistry. And that is the structure of today's presentation. First, we'll examine the primary molecular targets and receptor interactions. Then we'll move to network-level brain effects, perceptual mechanisms, motor control impairments, and finally, additional bodily effects and pharmacokinetics. By the end, you'll see how this one compound and its related tryptamines may redefine how we understand autism. When we talk about DMT's impact in the brain, it comes down to two primary molecular targets, receptors that shape everything downstream. The first is the serotonin 5-HT2A receptor. DMT is a potent agonist here, binding with high affinity. These receptors are densely expressed on layer 5 pyramidal neurons in the cortex, especially on their apical dendrites, which serve as integrators of cortical signaling. When DMT activates these receptors, it ramps up cortical excitability and enhances glutaminergic signaling. In other words, it pushes the brain into a hyperconnected, hyperexcitable state. The second is the sigma-1 receptor. DMT shows measurable activity here as well. Sigma-1 receptors modulate ion channels, influence neuronal excitability, and play a crucial role in regulating cellular stress responses. Under conditions of stress or hypoxia, sigma-1 signaling provides cytoprotection, essentially shielding neurons and keeping them functional. The net effect of these interactions is profound. Together, they trigger surges in cortical activity and radically altered signal processing, the very foundation for the perceptual, cognitive, and motor changes that define the DMT experience. Now, imagine if this chemistry were not a fleeting pharmacological event, but a chronic dysregulated state inside the brain. That possibility is exactly what we're considering in autism. Now let's move from molecules to networks, the level where brain-wide dynamics start to explain behavior. EEG and functional MRI studies show that within minutes of DMT administration, there are large reductions in alpha and beta rhythms, the very frequencies that normally keep brain activity structured and predictable. At the same time, there's a surge in signal diversity and entropy across brain regions. In other words, the brain becomes less ordered, more chaotic, but also more flexible. These changes correlate directly with the intensity of visual and immersive experiences reported by subjects. Functionally, this translates into widespread reconfiguration of connectivity. Networks that are normally segregated, like visual, auditory, and associative circuits, begin cross-talking in unusual ways. Constraints are loosened, creating a state of hyperconnectivity. Another critical piece is the thalamus, the brain's central gatekeeper of sensory information. Under DMT, the thalamus allows far more raw sensory data to flood the cortex. Filtering is diminished, integration is altered, and thalamocortical patterns are reshaped. Imagine the sensory world no longer being filtered down, but instead crashing into consciousness in full resolution. The impact on information processing is profound. Communication between networks increases. Bottom-up sensory signals dominate over top-down predictions, and the brain begins to recognize patterns it would normally ignore. This creates the conditions for novel perceptions, heightened sensory awareness, and at times overwhelming immersion. Now ask yourself, doesn't that sound strikingly similar to what we see in autism? Heightened sensory input, altered connectivity, bottom-up overload, all features that could be explained if the brain were living in a chronically dysregulated psychedelic state. One of the most powerful frameworks to explain what psychedelics do to perception is the REBUS model, short for Relaxed Beliefs Under Psychedelics. Under normal conditions, our brain relies heavily on high-level priors, top-down predictions that filter and constrain how we interpret sensory input. This is what gives us stability and coherence in perception. But under psychedelics, those priors are relaxed. Association networks become more flexible, less constrained, and bottom-up sensory data floods in with far greater influence. The result is a shift in the balance of perception. Internally generated imagery becomes vivid. Sensory modalities begin to crosstalk, producing synesthesia, like hearing colors or seeing sounds. Temporal processing warps, leading to distortions in the experience of time, and immersion deepens with an overwhelming sense of presence or connectiveness. EEG and functional MRI studies confirm this. As priors relax and entropy in brain activity increases, perceptual systems reorganize. Now consider autism through this lens. If endogenous psychedelics are chronically elevated, the REBUS model could help explain why individuals with autism often perceive the world in ways that seem radically different, why sensory experiences can be overwhelming, why time and rhythm feel altered, and why unusual cross-model associations emerge. It suggests that autism may not simply be a deficit of processing, but a fundamental shift in the predictive coding of the brain. So far we've looked at perception and cognition, but let's now consider motor control. Under DMT, movement itself becomes impaired, and the neurobiology gives us a clear explanation. First, the basal ganglia. These circuits rely on serotonergic modulation for smooth motor control. When 5-HT2A receptors are activated, motor command processing and proprioceptive feedback become distorted. The cortico-striato-thalamocortical loop is disrupted, breaking the precision of normal motor signaling. The substantia nigra is also affected. Experiments show that 5-HT2A activation alters firing patterns here, changing the balance between excitation and inhibition in circuits responsible for movement initiation and suppression. In effect, the system becomes unstable. The thalamus adds another layer. When thalamomotor connectivity is altered, sensory filtering of movement feedback is disrupted. This affects not only how movements are generated, but also how they are fine-tuned and corrected in real time. Clinically, this manifests exactly as you would expect. Tremors, ataxia, slowed movements, erratic or poorly coordinated motions. Under the influence of exogenous DMT, these effects are temporary and fade as the compound is metabolized. But imagine if endogenous psychedelics like DMT or Bufotenin were chronically elevated. Motor symptoms, tremors, awkward gait, impaired coordination would not just be transient, it could become a persistent part of the neurological phenotype. And here, again, we see a striking parallel in autism. Beyond perception, cognition, and motor control, DMT also produces striking bodily effects. Let's start with nausea. Serotonin acting on 5-HT3 receptors in the brainstem triggers vagal pathways, which drive the urge to vomit. This effect is particularly pronounced with ayahuasca, where DMT is combined with harmful alkaloids, amplifying serotonergic stimulation of the gut-brain axis. The autonomic nervous system is also powerfully affected. DMT can alter heart rate, often increasing it, and modulate blood pressure through broad serotonergic effects. Temperature regulation shifts, pupils dilate, and the entire balance of autonomic tone is reshaped. These changes demonstrate that DMT is not just a compound of the mind, It influences the body at a systemic level. Finally, sigma-1 receptor activity adds another dimension. By modulating ion channels and promoting protective signaling under hypoxic stress, sigma-1 receptors may buffer neurons from damage. In vitro, these pathways show protective effects that could influence whole body physiology. Now, it's important to emphasize, while these effects contribute to the total DMT experience, the hallucinogenic phenomena we've been discussing, the altered perceptions, the connectivity, and the motor changes, are primarily driven by 5-HT2A receptor activity. But these bodily effects remind us that endogenous psychedelics do not act in isolation. They reverberate across the brain-body axis, influencing everything from the gut to cardiovascular tone, and this may help us explain certain systemic features we see in autism as well. So let's bring it all together. At the receptor level, DMT exerts its strongest effects through 5-HT2A activation on cortical pyramidal neurons, driving excitability and altering signaling. Secondary sigma-1 receptor activity contributes to ion channel modulation and may provide neuroprotective effects, shaping the physiological landscape. At the brain-wide level, DMT reorganizes networks. Alpha and beta rhythms collapse, entropy rises, and connectivity patterns loosen. The thalamus allows more raw sensory information to pass to the cortex with far less filtering. Perceptually and behaviorally, this creates a very distinct state. The REBUS model helps explain it. Top-down priors lose precision while bottom-up sensory inputs dominate. Meanwhile, motor circuitry, modulated by the basal ganglia and the thalamomotor loops, become unstable, leading to impaired coordination, tremor, or slowed movement. The integrative understanding is this: DMT's effects are not random. They arise from a cascade that begins with specific receptor interactions, ripples outward through brain networks, and culminates in profound changes to perception, cognition, and motor control. Now imagine if this state were not brief, as with exogenous DMT, but chronic, driven by endogenous psychedelics within the developing brain. That is the radical possibility we must consider for autism, a neurodevelopmental condition shaped not just by genes or environment, but by the very psychedelic chemistry inside of us. Here are the key studies underpinning this presentation for DMT. On the left, you see the primary receptor and mechanistic work, 5-HT2A and sigma-1 receptor activity, cortical excitability, and pharmacokinetics. On the right, network level and clinical studies including functional connectivity, the REBUS model, and physiological profiles. Together, these represent the peer-reviewed foundation for everything we discussed so far. Now let's turn to Bufotenin, or 5-HO-DMT. This is another indole tryptamine, structurally related to DMT and found both in plants and in animal sources like the Sonoran desert toad, which secretes it from its skin glands. Mechanistically, Bufotenin is fascinating because it's a non-selective serotonin receptor agonist, interacting not just with 5-HT2A, but also with 5-HT1A, 5-HT2C, and 5-HT3. That means its influence spans mood regulation, perceptual distortion, sensory gating, and even nausea pathways. At the network level, Bufotenin alters cortical connectivity and thalamic gating, very much like DMT, opening the door to unusual sensory experiences and hallucinations. On the body side, it carries cardiovascular effects and potential therapeutic properties that are still being explored. So in this section, we'll follow the same structure as with DMT: molecular targets, network effects, perceptual and motor changes, and systemic impacts, building a comprehensive view of how Bufotenin may shape brain and behavior. Bufotenin, or 5-HO-DMT, is a close biochemical cousin of DMT. differing only by a hydroxyl group on the indole ring. Despite that small structural change, its pharmacology is remarkably broad. It acts as a non-selective serotonin receptor agonist, targeting multiple receptor subtypes simultaneously. At 5-HT2A receptors, it drives visual hallucinations and altered perception, similar to DMT and LSD. At 5-HT2C receptor, it modulates dopamine and norepinephrine release, shaping emotional and anxiogenic responses. Through 5-HT1A, it influences global serotonergic tone, anxiety modulation, and even thermoregulation. And uniquely, through 5-HT3, it acts directly as an ion channel, mediating nausea and vomiting pathways. At the cellular level, bufotenin enhances cortical excitability and glutaminergic signaling. Interestingly, there is also evidence for potential anti-inflammatory and analgesic effects, reminding us that these molecules are not just psychedelic, they engage deeply with cellular signaling cascades. In short, bufotenin doesn't act on one switch in the brain. It pulls multiple levers at once, spanning perception, emotion, sensory filtering, and body regulation. That makes it a particularly intriguing candidate when we consider autism as a condition of altered serotonin and indole-tryptamine metabolism. At the network level, bufotenin appears to behave much like DMT, though fewer direct neuroimaging studies exist. Based on its structure and receptor profile, we can predict several key effects. First, neural activity patterns. Bufotenin likely disrupts cortical oscillations in the alpha and beta bands, the very rhythms that organize perception and cognition. Signal complexity and entropy increase, reflecting a loosening of order and a rise in chaotic connectivity. And because it acts broadly across serotonin receptors, predictive coding in sensory systems is destabilized. Bottom-up sensory data begins to dominate over top-down expectations. Connectivity is altered in parallel. Thalamocortical gating opens up, allowing more raw sensory input to flood into cortical regions. The integrity of the default mode network is likely disrupted, a hallmark of the psychedelic state. Communication increases between networks that are normally kept separate, producing crosstalk and unusual associations. Even in animal studies, these connectivity changes correlate directly with visual processing distortions. Clinically, the result is predictable: visual hallucinations, synesthetic experiences where senses blend together, and distortions in time perception linked to disrupted synchronization of neuronal networks. So while bufotenin is less studied than DMT, everything we know about its structure and receptor activity points to comparable, if not broader, network effects that could deeply reshape perception and sensory experience. Bufotenin's broad serotonergic activity translates directly into altered perception and emotion. On the sensory side, non-selective 5-HT2A and 5-HT2C receptor activation disinhibits association networks and disrupts normal predictive coding in the visual cortex. High-level perceptual priors lose their grip, while bottom-up sensory inputs take over. This produces intensified visual processing, brighter colors, sharper patterns, and a weakened boundary between internal imagery and external perception. Altered thalamocortical gating ensures more raw sensory data reaches awareness, fueling visual hallucinations. On the emotional side, 5-H21A and 5-HT2 receptor activation modulates limbic circuits. Amygdala reactivity shifts, changing the salience of stimuli, what feels threatening, safe, or overwhelming. And when sensory modalities blend, synesthesia can occur, where sounds are seen or colors are felt. The overall effect is a distinct perceptual state: visual hallucinations, altered bodily awareness, emotional reactivity, and cross-modal sensory blending. Bufotenin's wide receptor profile makes it particularly potent at reshaping both perception and emotion simultaneously. Bufotenin, like DMT, also exerts powerful effects on motor control. Neurologically, this happens through serotonergic modulation of basal ganglia circuits across multiple receptor subtypes, 5-HT2A, 2C, and 1A. These receptors are embedded in the corticostriato-thalamocortical loops that govern movement coordination. When disrupted, fine-tuned motor signaling collapses. In the substantia nigra and related motor nuclei, altered firing patterns interfere with the initiation and control of movement. Because bufotenin binds broadly across serotonin receptors, its motor effects are even more extensive than more selective psychedelics. Its 5-hydroxy group slightly shifts its receptor binding profile, adding another layer of influence on motor regulation. Clinically, this translates into very recognizable manifestations: difficulty with fine and gross motor coordination, unsteady gait or impaired balance, the perception of bodily heaviness or altered proprioception, and erratic or slowed movements during peak exposure. Importantly, these effects are route dependent. With higher central bioavailability, for example, intranasal versus oral delivery, motor impairment becomes much more pronounced. So bufotenin, though its wider receptor reach, not only alters perception, but also deeply impacts motor systems, producing effects that echo what we see in disorders characterized by dyspraxia and motor instability. Bufotenin doesn't just alter perception and motor control, it also impacts the body more broadly. In the digestive system, its activity at 5-HT3 receptors in the brainstem triggers nausea and vomiting reflexes, similar to other tryptamines, but with a distinct twist due to its hydroxyl group. These GI effects are well recognized in both experimental models and traditional contexts. Cardiovascularly, bufotenin stimulates 5-HT4 receptors, increasing cardiac contractility. It also shifts blood pressure and heart rate through autonomic nervous system modulation. Even at low concentrations, studies in transgenic mouse models confirm direct impacts on cardiac function. What's surprising is that bufotenin may also have anti-inflammatory and analgesic properties. Through inhibition of the JAK2 STAT3 pathway and sigma receptor engagement, it shows potential protective effects in hypoxic or stress conditions, hinting at possible therapeutic applications beyond its psychedelic profile. And historically, bufotenin was first isolated from toad venom and has been used in traditional medicine for centuries. But it's a double-edged sword. Cases of toxicity from improper handling of toad secretions remind us how important it is to fully understand its physiological effects. So bufotenin is not only a psychedelic, it is a systemic compound touching the gut, heart, immune system, and beyond. That complexity makes it a powerful candidate for explaining multi-system features in conditions like autism. Finally, let's look at bufotenin's pharmacokinetics and metabolism. When taken orally, bufotenin undergoes extensive first-pass metabolism by monoamine oxidase, MAO, which rapidly breaks it down and dramatically limits its psychoactivity. This is why oral ingestion of toad secretion historically produced only weak effects. By contrast, intranasal or inhaled routes bypass first-pass metabolism, leading to far greater central bioavailability and much stronger psychoactive outcomes. Parenteral administration, through direct injection, produces the highest bioavailability, though this is rarely used outside of research. On the metabolic side, MAO is the key enzyme, degrading bufotenin through oxidative deamination. Structurally, bufotenin is simply a hydroxylated derivative of DMT, and it shares close biotransformation relationships with both DMT and 5-MeO-DMT, depending on methylation and hydroxylation processes. An important point here, MAO inhibitors can radically alter bufotenin's pharmacokinetics, preventing breakdown and significantly increasing both the duration and the intensity of the effects. So pharmacology determines whether bufotenin is barely active or profoundly psychoactive, and this metabolic fragility could explain why endogenous bufotenin, even at low levels, may still have disproportionate effects if MAO pathways are impaired or genetically variable. I have a separate slide for that coming up in regards to the MAO genetic variations we see in autism. To summarize, bufotenin acts broadly across serotonin receptors, disrupting cortical rhythms, increasing entropy, and altering thalamocortical gating. Clinically, it produces hallucinations, motor impairments, and cardiovascular changes, but may also carry therapeutic properties. Importantly, its effects are highly dependent on route of administration. Future research will need to map receptor-specific contributions and validate these network changes in humans. Here are the key references supporting bufotenin research. On the left, we have studies on receptor binding and molecular mechanisms, including its broad serotonergic profile and cardiovascular effects. On the right, biological and clinical findings, animal models, anti-inflammatory pathways, and evidence of elevated bufotenin in patients with mental illness. Together, these works form the scientific foundation for considering bufotenin as a significant player in brain and systemic physiology. This slide ties everything together by showing how these compounds emerge from the same biochemical origin, tryptophan. On the left, the well-known pathway converts tryptophan to serotonin, then to melatonin, regulating mood and circadian rhythm. In the middle, tryptophan can instead be decarboxylated to tryptamine, and with the enzyme INMT, a methyltransferase, converted into DMT. On the right, a similar route produces serotonin, which can then be methylated to form bufotenin or 5-HO-DMT. The point here is simple but profound. Serotonin, melatonin, DMT, and bufotenin are not separate worlds. They are parallel branches of the same tree, all derived from tryptophan. Which branch is emphasized may depend on genetics, enzyme activity, or environmental triggers. And if the psychedelic branches of this tree are overactive, producing DMT or bufotenin in excess, it could shift brain development toward a state we recognize as autism. This slide shows how endogenous compounds like DMT and bufotenin map onto the same receptor landscape as classic psychedelics. Notice that DMT and bufotenin have strong agonism at 5-HT2A receptors, the same primary target as LSD and psilocybin, which are known for radically altering perception. Bufotenin also acts on the 5-HT1A, 2C, and 3 receptors, giving it broader systemic reach. Both compounds engage sigma-1 receptors, which may add stress-modulating and cytoprotective effects. Compare this to serotonin, which sits at the center of normal signaling, and to exogenous drugs like LSD, psilocybin, and ketamine. The overlap is striking. It means that the same receptors driving psychedelic states are accessible to our own endogenous chemistry. The key takeaway here, autism may not be explained by a serotonin imbalance alone, but by dysregulation of an entire psychedelic receptor network inside the brain. This systematic review highlights one of the most consistent biochemical findings in autism, hyperserotonemia, or elevated blood serotonin. Out of over 1,000 publications screened, 59 studies confirmed higher serotonin levels in individuals with autism. But here's the key. Serotonin is only one branch of the tryptophan pathway. If serotonin is elevated, it raises the question, what happens to its metabolites downstream? Could the excess serotonin be feeding into the production of indole tryptamines like DMT and bufotenin? This is where my hypothesis builds on established science. Autism has long been linked to serotonin abnormalities. What are we now proposing is that the story doesn't end with serotonin. It extends into an entire psychedelic chemistry inside the brain. This paper from the University of Pavia in Italy is a landmark. For the first time, bufotenin, the same compound found in toad venom, was shown to be elevated in humans with autism and schizophrenia. Urinary bufotenin levels were more than twice as high in ASD patients compared to controls. Even more striking, these elevations correlated directly with hyperactivity scores in autism. The conclusion is inescapable. An endogenous psychomimetic molecule, structurally similar to known psychedelics, is measurably elevated in autistic individuals. This is no longer theoretical. We're now looking at hard biochemical evidence that the psychedelic chemistry of the brain may be part of the autism phenotype. This slide makes the pharmacology very clear. Here we see the relative receptor binding affinities of the main tryptamines. Serotonin itself binds strongly to 5-HT1A and 2A receptors, setting the baseline for normal signaling. Bufotenin, however, shows even stronger affinity at the 5-HT2A, the same receptor that drives the psychedelic effects of LSD and psilocybin, and it also engages sigma receptors, adding stress response and perceptual effects. DMT similarly binds to 5-HT2A and sigma-1, reinforcing its dual psychedelic and neuromodulatory role. Tryptamine, the simple precursor, has only weak receptor activity, but it serves as the gateway molecule. The key point: serotonin, bufotenin, and DMT are not just structurally related, they are pharmacologically linked through overlapping receptor targets. This means that a shift in metabolism from serotonin towards bufotenin or DMT could transform normal neurotransmission into a psychedelic state inside the developing brain. This brings us to the connection with current westernized autism treatment. The two FDA-approved drugs for autism irritability, risperidone and Abilify, both act on the same receptors targeted by endogenous tryptamines. Here you see the receptor affinity profiles. Risperidone binds strongly to the 5-HT2A and dopaminergic D2 receptors. Abilify does as well, but with even stronger affinity for 5-HT1A. Why is this important? Because 5-HT2A is the main psychedelic receptor, activated by DMT, bufotenin, LSD, and psilocybin. 5-HT1A shapes emotional regulation, while dopaminergic D2 integrates motor and reward pathways. This slide captures what happens when DMT and bufotenin are not released in sudden peaks, but instead persist chronically at low levels, the very scenario that could redefine autism as an endogenous psychedelic state. Bufotenin, with its high affinity for 5-HT2A and 5-HT1 receptors, produces subtle but pervasive effects: cognitive fog, head pressure, hyperactivity, anxiety, and OCD-like behaviors. Sensory sensitivity emerges, light tolerance, afterimages, occasional synesthesia. On the physical side, we see vasoconstriction, flushing, tachycardia, nausea, and skin tingling. DMT at low levels brings its own profile: dreamlike thinking, time perception shifts, heightened imagery, emotional liability, and sleep disturbances with vivid or lucid dreams. Physiologically, mild pupil dilation, tremors, and heart rate fluctuations appear. Taken together, chronic low-dose exposure does not resemble a psychedelic trip. It resembles a developmental disorder. The very symptoms we label as autism could instead be the neurobehavioral fingerprint of sustained low-level psychedelic chemistry inside the brain. This slide reveals the missing piece of the puzzle, how endogenous psychedelics actually form inside of us. It starts with tryptophan, a dietary amino acid. Gut bacteria step in, decarboxylating it into tryptamine, a process already shown in Clostridia, Ruminococcus, and other species. From here, the host takes over. On the DMT pathway, the enzyme INMT, which is a methyltransferase, sequentially methylates tryptamine into N-methyltryptamine and then into DMT. On the Bufotenin pathway, tryptamine undergoes hydroxylation into serotonin, and further N-methylation yields Bufotenin. This is a bacterial-host collaboration. The microbes provide the raw tryptamine, and human enzymes finish the job, producing DMT and Bufotenin without any problems and within our tissues. The implication is profound. Autism may not just be a genetic or neurological condition, but a microbiome-driven psychedelic state, where bacterial metabolism directly fuels the brain exposure of these powerful tryptamines. Here we see the microbial side of the equation, bacteria with the enzymatic machinery to make psychedelic chemistry possible inside the host. On the left, species like Streptomyces, Pseudomonas, Clostridium, and Burkholderia are proven methyltransferase producers, capable of generating methylated indole derivatives. These enzymes function much like the human INMT, and under the right conditions could support endogenous DMT synthesis. In the center are bacteria carrying tryptophan decarboxylases, enzymes that convert tryptophan into tryptamine. These species include Clostridium, Ruminococcus, Morganella, Klebsiella, and even common gut residents like E. coli and Lactobacillus. On the right, we have the suspects: Prevotella, Disulfibrio, Blaudia, Doria. These are often elevated in autism microbiomes and are correlated with altered tryptophan metabolism or methyl donor cycling. Taken together, this is the foundation of a host-microbe collaboration. The microbiome not only modulates serotonin, as we discussed, but may also be feeding the brain a steady trickle of endogenous psychedelics. DMT is not limited to surface serotonin receptors. Because of its small lipophilic structure, it freely diffuses across neuronal membranes and activates intracellular targets. One of the most important of these is the sigma-1 receptor, located in the endoplasmic reticulum and mitochondria. Activation here regulates calcium signaling, reduces cellular stress, and promotes dendritic growth and synaptic connectivity. Psychedelics like DMT have been shown to increase neurite outgrowth, spine density, and functional synapses, clear markers of neuroplasticity. This is more than a hallucinogenic activity. It represents a fundamental reshaping of brain circuits. When we look at autism, one of the most consistent neuroimaging findings is hyperconnectivity in cortical association networks. If endogenous DMT is elevated in autism, its intracellular actions may help explain this phenomenon, driving excessive wiring, abnormal network integration, and altered information flow. This slide highlights the consequences of slow or low activity variants of the MAOA gene. Individuals who carry this version of the gene metabolize tryptamines less effectively, which means neurotransmitters like serotonin and dopamine can build up, especially under stress or when extra substrate is present. Functionally, this is linked to poor impulse control, greater risk of aggression, and heightened sensitivity to environmental adversity, including altered responses to serotonergic drugs and psychedelics. We also know that congenital MAOA deficiency produces a striking clinical picture called Bronner syndrome.
marked by impulsivity, aggression, and mild intellectual disability. Beyond that, studies have shown that downregulation of MAOA in the placenta correlates with adverse neurodevelopmental outcomes and altered infant temperament. Importantly, this gene is also highly sensitive to environmental interaction. Carriers of MAOA in their slower form are particularly vulnerable to the effects of early life stress and trauma, as demonstrated in classic studies by Caspi and Kim Cohen. I would add here that in my own work I too often see MAOA genetic downregulation, and this carries extra weight because of the gene's biology. MAOA is located on the X chromosome, so it is essentially hemizygous in males, meaning boys only have one copy. If that single copy is a slow-acting variant, there's no backup. Girls, in contrast, have two copies and some degree of protection. This X-linked difference likely contributes to sex-based disparities and vulnerability to aggression, impulsivity, and perhaps responses to tryptamine-based neurotransmitters and psychedelics. We're now taking this bold hypothesis into action. Together with Stanford, UC Davis, and Autism Biomedical, we're designing the first ever study to measure the complete psychedelic metabolome in autism across stool and plasma in 90 individuals. This has never been attempted, not in autism, not in psychiatry at all. By mapping endogenous tryptamines, we may uncover a unifying biochemical pathway that explains extraordinary perceptual phenomena in ASD. We're seeking funding partners who recognize the paradigm-shifting potential of this work. If you want to help us rewrite the story of autism, here is your chance. Please contact me at cbogner@drbognerhealth.com, and thank you so much for your time, and I hope you enjoyed this brief summary.